Hello, everyone. So our research title is PXR activity in breast cancer cell line. And so the purpose of our experiment was to study endogenous PXR expression across different cancer cell lines. The cell lines that we used is HEVG2, which is a hepatocellular carcinoma, LS180, which is a colorectal adenocarcinoma, P47D, it's a breast ductal carcinoma that is estrogen positive, progesterone positive, and HER2 negative. MCF7 is a breast adenocarcinoma, which is also HER2 negative. MBA-MB231 is a breast adenocarcinoma that is triple negative. So now that we know the cell lines that we are going to use, I'm going to move on to talk about our lab methods. So first, we harvested these cell lines and we lysed them to extract the protein, and then we ran that protein in 10% SDS page to make sure the proteins separate based on their size. And then we ran a Kumasi blue stain, which would serve as our loading control to make sure we have the same amount of protein across the different wells. And as we can see here, they look relatively similar, but with the MCF7, we do see a band over here, which we think to be a mutated B53, which is around 43 kilodalton. And then we also did a Western blot, and we probed with anti-PXR monoclonal antibody. And HEVG2 and LS180 serve as our positive control because we do know that they express endogenous PXR. And as we can see here, there's a band right here, a little bit over 55 kilodalton. P47D also express the same band. So in conclusion, we know that HEVG2, LS180, and T47D express endogenous PXR. From our cancer breast cell lines, T47D is the one that expresses endogenous PXR. So we decided to study PXR activation by different ligands in transfected T47D cells using gene reporter assay. So Jack already talked about this, but just to refresh your memory, the reporter gene assay has a xenobiotic response enhancer module, which has endogenous RXR and exogenous PXR. So when a PXR activator, such as rifampin or SR12813, bind to PXR and activate it, we get a luciferase activation. So now that we reviewed our gene reporter assay, we'll go take a look at our data. So these are the ligands that we used. We use vehicle rifampin, which is an antibiotic that is usually used to treat tuberculosis, and SR12813, which is a failed cholesterol-lowering drugs. These are our cofactors. And from our data, we see that without PXR, whether we had cofactors or without cofactors, we see little activation of PXR by these ligands. And in the presence of PXR, we see that both drugs or ligands activated PXR. And when we added the cofactors, that activation was further magnified. So now I'll hand it off to Brendan, who will be talking about real-time qT-PCR data.
Thank you, Yasmeen. So as you said, I focused a lot of my time doing real-time qPCR. The first goal we had was to determine if the T47D cells, as a reminder, ER positive, PR positive, HER2 negative, if they had endogenous PXR and endogenous PXR activity. So we got the same ligands, rifampicin and SR, put them in T47D cells and went to see if we had the correct target gene induction, such as CYP3A4 and UGT1A1. Our results for that, we have our real-time qPCR over here. We have our actin as a control, and we have our CYP3A4 in blue. As you see, there's our vehicle line, and then our rifampicin and SR lines are almost perfectly overlapping. And as you can see, there's a clear leftward shift with the rifampicin SR showing us that there is induction of these when that CYP3A4 is induced when given rifampicin and SR. To further prove this, we ran a delta-delta CT on this data, showing that we have a 1.4 and 2.1 fold induction of CYP3A4 when exposed to rifampicin and SR. We ran this again with UGT1A1, another PXR target gene, and we had very similar data. We have a 1.5 and 2.6 fold induction of UGT-A1A when exposed to rifampicin and SR. The second goal we had with doing real-time qPCR was to explore student Dr. Jack Hemstaff's discovery about PPAR gamma and his LS180s. We wanted to see if this PPAR gamma finding was similar in T47D cells. So we ran a real-time qPCR with rifampicin and SR, and instead of just PPAR gamma, we ran a bunch of genes that are very similar to it as PDK4, PLIN2, and PLIN4, and our results are nothing. There is no statistical significance of any kind of induction or repression with these genes, showing us that the finding that Jack had with the LS180s is something unique to his LS180 colon cancer cell line. It is not present in the T47D breast cancer cell line. This could be due to a variety of factors, such as either little or absent PPAR gamma in the T47D cells, or the PPAR gamma is somehow repressed in these T47D cells. You can do thanks.
All right, and we would like to conclude by thanking our mentor, Dr. Jeff Spottinger, Dr. Bradley Creamer, Dan Probst, and Catherine, as well as the NIH grant and our amazing lab mates, Jack, Abby, and Chi. We couldn't have done it without you, and it was the most amazing experience ever. Thank you all.